Hello and welcome everyone. So in the last class we learned about DNA methylation. So whenever there are CpG dinucleotides on the DNA, those get methylated and it leads to gene silencing. So today we'll be focusing on the mechanism of repression via methylation. So how does methylation actually leads to the gene silencing? So whenever there are CpG dinucleotides, DNMT can bind to those CpG dinucleotides and it methylates the cytosine. Okay, it methylates the fifth 5 carbon of the cytosine. And due to that, cytosine get converted to 5 methyl cytosine. So after the DNMT binding, DNA gets methylated. So this methylated DNA, this is represented like this. Okay, so this methylated DNA is now capable of binding certain proteins, and those proteins are known as methyl binding proteins. Methyl Take a note that methyl binding proteins can only bind to methylated DNA. Okay, and that's why they are known as methyl binding proteins. So there are two types of methyl binding proteins. Okay, MECP1 and MECP2. So MECP1 means methyl cytosine binding protein 1, while MECP2 means methyl cytosine binding protein 2. So this is those two types of proteins have different conditions for binding. So MECP1 needs a threshold number of methylated CpG bind. Okay, means there should be there should be a threshold number of methylated CpG. Only if those much CpGs, methylated CpGs are present, then only MECP2 is able to bind to it. While MECP1, while MECP2 can bind to single methylated CpG. Okay, so MECP2 has the ability to even bind to the single CpG, while MECP1 can only bind if there is certain a threshold is reached, and that's why MECP2 binding is often seen in CpG-rich islands. Okay, why? Because only CpG-rich islands are able to reach to that threshold number, right? While MECP2 binding is often seen at CpG-poor islands. Okay, so those MBPs, those methyl binding proteins, they help in the recruitment of HDACs. So what are HDACs? HDACs are histone deacetylases. So the substrate is histones, means they work on the histone, and what does it do? It deacetylases the histone, means it removes the additional acetyl group which is added on the N-terminal tails of histone. So what does histone deacetylase do? They perform the histone deacetylation, means the removal of acetyl group, and removal of acetyl group results into chromatin condensation. How does it do that? Let's have a quick look at it. Okay. So in the previous video lectures, we have learned that DNA is wrapped up around the histones and it forms nucleosomes, which are the basic structural unit of the chromosome. Okay, so histones, they are positively charged proteins, while DNA is negatively charged. Okay, and this is how the interaction between both of them is stabilized. So whenever there is addition on the addition of the acetyl group on the histone tails. Okay, histone. I hope you remember that histones have histone full domain, and N-terminal tails as well as C-terminal extensions. Okay, so those N-terminal tails are very crucial for the compaction of DNA. Okay, and on the all of the modifications, all of the post-translational modifications are done on the histone tails as well as histone cores. Some of them are also done on histone cores. So whenever there is addition of acetyl group on the histone tail, we call that modification as histone acetylation. And due to addition of acetyl group, this positive charge is neutralized. Okay, and due to neutralization of the charge, what happens is that if there is no positive charge on histone, the DNA histone interaction, this interaction will become weak. And because this interaction becomes weak, the chromatin conformation becomes open. So due to open configuration of the chromatin, now the DNA is more accessible, right? And this more accessible DNA leads to the gene expression. Okay, so whenever, so this is so means histone acetylation is an active mark. Why? Because active mark. Why? Because it is leading to the gene expression, right? Because whenever there is acetylation, it will weaken it will weaken the DNA histone interaction, and due to that, it will lead to gene expression. That's why whenever there is histone acetylation, we call it as an active mark. So what happens that MECP2, they recruits histone deacetylases, means they do the histone deacetylation, which will lead to closed configuration of chromatin. Okay, means it will compact the chromatin further, it will lead to chromatin condensation, and that is why now gene will be repressed. This is how gene will be repressed. Okay, so this is the mechanism of gene repression via methylation. I hope this is clear to you all.